Welcome to this presentation on 1D to D3D and multidimensional signals and images in medical imaging. This video is presented by Professor Gottapu Sasibhushana Rao. In this presentation, we will understand how signals and images are represented mathematically in 1, 2, and higher dimensions, and then see how computers represent and process medical images using numerical pixel values. Two fundamental concepts in signal and image processing are signal and system. A signal is a measurable quantity that varies with an independent variable, such as time or space, and carries information. Mathematically, it can be represented as a function. In the figure, the horizontal axis represents time, and the vertical axis represents signal amplitude. At any instant, the function gives the corresponding amplitude value of the signal. The waveform shown is periodic, meaning that it repeats after a fixed time interval, called the period. So, a signal may be viewed as information expressed mathematically as a function of one or more independent variables. A system acts on this signal to produce an output, which will be introduced in the later slides. Continuing from the previous slide, a signal is represented mathematically as a function of an independent variable. The measured quantity may be voltage, pressure, intensity, or any other physical parameter, and its value changes with time or space. In the figures, the waveform is shown as a function of one independent variable. The horizontal axis represents the independent variable, such as time, while the vertical axis gives the corresponding signal value or amplitude. For example, at a particular value x1, the function f, x1, gives one specific measured value. The sine wave figure illustrates this clearly. As the independent variable changes, the signal value also changes continuously. Since only one independent variable is required to describe the signal, it is called a one-dimensional, or 1D signal. So, the dimension of a signal is determined by the number of independent variables, not by the number of axes used to draw the waveform. This idea leads directly to 2D, 3D and multidimensional signals and images in the next slide. This slide shows how signals are classified by the number of independent variables. A 1D signal depends on one variable, such as time, f, t, as in audio, ECG, or EEG. A 2D signal or image depends on two spatial variables, f, x, y, as shown by the rectangular image plane. Examples include grayscale images, X-rays, CT slices, and Mr. slices. The video sequence shown in the figure introduces time as an additional variable, giving f, x, y, t. Similarly, a 3D medical volume can be represented as f, x, y, z. For dynamic 3D MRI or 4D CT, the data become multidimensional, for example f, x, y, z, t. The image examples at the bottom also show that digital images are represented using pixels, with different bit depths determining the number of intensity or color levels. This slide presents examples of one-dimensional and two-dimensional signals used in biomedical and imaging applications. The ECG waveform represents the electrical activity of the heart as voltage varying with time. The EEG traces similarly represent electrical activity of the brain as time-varying signals. The bold signal shows how the measured fMRI response changes across successive scans. These are all examples of 1D signals, because each depends on a single independent variable. The MRI and PET images shown in the lower part are examples of 2D signals or images, where intensity varies with two spatial coordinates, x and y. This slide introduces the concept of a system. A system performs an operation or transformation on an input signal and produces an output signal. It may be implemented using hardware or software. In the first figure, the input f, kiss smiley is applied to the system S, producing the output G, x, is equal to S, f, kiss smiley. The medical imaging example shows this idea in stages. The PET scanner acquires measured data, a reconstruction system converts that data into images, and an analysis system further processes the reconstructed images to obtain useful information, such as a time activity curve. Thus, a signal is a function, while a system is an operator acting on that function. This slide explains the working principle of positron emission tomography, or PET. A radioactive tracer is introduced into the body, and the emitted positron interacts with an electron. This produces an annihilation event, generating two gamma photons that travel in nearly opposite directions. As shown in the figure, the ring of detectors detects these two gamma rays simultaneously. From many such detection events, the PET system estimates the tracer distribution inside the body and reconstructs a functional image. So, PET converts detected gamma-ray events into spatial information about physiological activity. No of detectors maps the destination of these chemicals in the brain by sensing the emission of radiation in the form of gamma rays. This slide summarizes the complete PET imaging process. First, a radioactive tracer is injected into the patient. The emitted positron interacts with an electron and produces an annihilation event, generating to 511 keV gamma photons traveling in nearly opposite directions. As shown in the detector ring figure, these photon pairs are detected simultaneously by opposite detectors. A large number of such coincidence events are collected by the PET scanner. The measured data are then processed and reconstructed to form the final PET image shown on the right. Thus, PET imaging follows the sequence: tracer injection, positron emission, annihilation, photon pair detection, data acquisition, image reconstruction.